So, hello everybody. Thank you for attending this fantastic session. Just two things for our esteemed speakers. We are happy to have 17 companies today. You have 3 minutes to present, and please also then leave about 2 minutes for questions and answers. I would really like to ask you to keep the time. And for our audience, thank you for being very attentive, and you can support the companies by asking questions. And I would really like to ask you to do that. So with that, our first speaker is Christian Berger, who is CEO of ANNIMO Health. And this is an interesting case where some of the immuno-oncology and vaccination therapies goes into expensive animals.
Thank you very much, Tamas and team, for the invitation, the honor to open the session and kind introduction. Thank you to listen to ANNIMO Health, which means immuno-oncology in animals, ANNIMO. We own a technology that we believe is from human health breakthrough and leading edge in modern immuno-oncology. Actually, the technology comes from vaccines combined with a backpack or smartest possible immunostimulation intracellular. The key differentiator is the cells do not learn. They die and they do not learn. The neighboring cells do not learn that their neighboring cells are dying. Early testing for humans was done in phase 2, now in human, was done in horses and animal health. This is the patent box actually we took, and we built a very nice risk-reduced business case for horses, camels, dogs, and cats later on. Let's go into horses. 11 to 12% of all horses today worldwide suffer from skin cancers. They're called sarcoids. There are 7 classes. And you see the pictures here. They can be devastating, normally in soft tissue in the genital area, with the neck basically, or with the eyes. The medical need is very high. There's no therapy or cure. You can do a bit of surgery. You can give a little bit of photodynamic radiation. You can try some chemotherapy, but basically the horse is destined to slowly die. For the owners, it's a financial wipe-off of the value of the horses. You have high therapy and care costs, and emotionally it's very hard for the owners to see the horse suffer and die. With that superior technology, we believe, we were just testing for safety to go into human. And just 2 to 3 injections basically meant these pictures of a couple of horses out of 29. Just injection, no surgery. Within 4 to 10 months, the melanoma, the sarcoids were disappearing. You see some others here, bleeding status, basically wiped away. The technology behind is very simple. It is that dual approach of having a triple viral vector going to the cancer cell, completely targeted only to the cancer cell, and inside stimulating interferon cascades, different classes of interferon, and the cell is dying from within inside the cell. There's not much signaling or not at all signaling to the outside world, which means the neighboring cells are not learning. That is probably the trick. We had just 29 horses treated in a very unsophisticated 2 or 3 injections, and we wanted to have safety for going into human. 22 were cured, complete response or response. Cured completely with complete response after 3 years still, not a single oncological blood parameter being elevated, completely wiped out. 14 out of 29, unsophisticated. We are now standing in front, having organized CMC production, doing phase 1 again with CMC accredited drug, doing 1 combined phase 2, phase 3, agreed with EMA and USDA, FDA. We want to focus on European launch, on US launch and Canada. LATAM is an upside, Asia Pac is an upside. We see the launch upgrade here, and we plan to introduce 28, 29. Financial model behind is solid. We are supported by UBS, New Innovators, and have modeled that out with Harvard Business School. And you see the dark parts of the bars, which is Europe, the main market, a little bit US. There's a lot of upside. We just need 1.2% of financially accessible horses in Europe and US to make that business case.
So, Christian, sorry to interrupt. Maybe a final statement.
Absolutely.
Before
Final
we need
statement.
to move on.
Safe investment case. The company valuation goes up. The risk is manageable. We have all the assets. This is the team. We will return the investment 4.3 times over 4 years. And I'm CEO. I'm also the project manager and the senior study nurse, so we can get execution right. When the strategy is clear, execution is the new strategy. After horses, we go camels, dogs, and cats, and we might even go back to human health to feed back what we have learned. Thank you very much.
The hard sequel. So Robert Langer was asked last year at an event, why don't we see cell and gene therapies flood the market? They promise to change the world. It's a delivery technology. We need a safe, universal delivery technology. Once we have that, we should see companies churn out 25 assets every year. So we have just that. We have a universal platform to deliver nucleic acid-based therapies where no one else can. We do it in a quite different way than you're probably used to seeing. So my background is electrical engineering. I did my PhD in wave matter interactions with electromagnetism. So our system uses an external system, creating electromagnetic waves that focuses the energy inside the body from the outside. In this focal point, you can sort of imagine you have a looking glass focusing the rays of the sun, and you can start a fire with that. Instead of starting a fire, we're electroporating the cells in this focal point. The cells are electroporated, and they can take up these nucleic acid-based therapies that cannot be taken up by the cells otherwise. This is not just a nice, fluffy idea from a desk. We have shown this. First proof point is our in vivo studies where we showed the high localization capabilities of our platform. So what we did, we injected naked pDNAs into these animals, and then we treated one thigh only after the injection. 48 hours after, we see the expression of the protein highly localized to the area that we treated and nowhere else. We are agnostic across different molecular entities. We've shown the pDNA expression, as you just saw. These are in vitro results. And we've recently also shown we can transfect Cas9 proteins and knock down genes with that. Again, let me emphasize, no chemical adjustments, no extra formulation to the Cas9 proteins, pure proteins. Also ASO uptakes, siRNAs, and even small molecules. We actually started with small molecules, but realized that the real value is probably in genetic therapies. We develop assets preclinically, and then we license them out. We intend to license them out for clinical development. We truly believe in the breadth of our platform because we can do any indications, any molecules. Sorry. I think I was one slide ahead. There we go. First program will be a pDNA expressing IL-2, very proven against solid tumors. But there are more indications where IL-2, again, highly localized expression of IL-2 will have a huge effect. And those are the next in our pipeline. The financial model, it will be an aggregation of the license deals. It will have a upfront cash component, and it will have the milestone royalty payments. We're a very broad team with a broad set of competencies. We have engineers, medical doctors, scientists, and biotech executives with decades of experience in the industry. We are currently looking for investors for a $5 million round. We already have commitment from existing investors. Plus 10 seconds. Thank you.
So, thank you, Niels. Maybe there are questions out there, please.
As your first asset is in the oncology space, how do you recommend targeting metastasis?
Great question. So what we do is highly localized, so it's not good for all the small metastases, the rest of the body. Our first indication, head and neck squamous cell carcinoma, is where the primary tumor is of the major concern. If you can wipe out the primary tumor, you're pretty good, and the studies show this. The second effect of this is that we're actually not just killing the tumor, we're making the immune system kill the tumor. And there are a lot of studies also showing quite good effects from the abscopal effect. So once you let the immune system take care of the tumor, it can clear the rest of the body.
Welcome everyone. Thank you for the introduction. So today, the success rate of candidate drug is approximately 10%. And the drug development costs more than 2 billion per molecule. We believe we can make it much better. How? By integrating the human body drug response early in the drug discovery process. Let's take a look. In our body, the organs communicate with each other constantly. This determines the efficacy and the toxicity of the molecules and also the metabolites. This is what we call the systemic drug effects. At FluoSphera, we developed a unique in vitro multi-tissue systems that is able to mimic the complexity of the systemic drug response. Briefly, we encapsulate human microtissues into color-coded capsules that we co-culture together so that we can recapitulate the communication between organs. After drug treatments, we leverage imaging so that we can evaluate the effect of molecules on each tissue types thanks to the color code. To illustrate the power of our technology, we took irinotecan. It's an anti-cancer drug with an indication for the colon carcinoma. It's a prodrug, so it means that without the metabolization by the liver, it's not active. So we need to recapitulate these models. In absence of the liver, we do not see any effects. In presence of the liver, we could reveal in green the effects. We could reveal the systemic efficacy on the colon, the selective efficacy on the colon over the breast, and also the toxicity that is associated on the liver. So we can recapitulate the communication between organs. With our technology, we developed a platform, FluoXcovery, to actually give a tool for the systemic in vitro drug discovery. We can evaluate the selectivity of a molecule, FluoSelect. We can evaluate at which concentration we should work to have efficacy over toxicity, the FluoScore. We can combine our technology with omics analysis to determine the mode of action and also biomarkers. To support our vision, we have a great team with my co-founder. We are the co-inventor of the technology. We have also seasoned entrepreneurs. We also have financials and a great scientific team and advisor. At the moment, we are raising 1.5 million to accelerate our growth and actually the generation of revenues. So if you are interested, come to us after this presentation. Thank you very much.
Thank you, Celia. Any questions from the audience? I personally would be interested in how many different tissues or cell types can you combine in a way that you can still draw conclusions of that?
Yeah, that's a very good question. So at the moment, in terms of multiplexing, we are able to mix 6 different tissue types together. Okay. So, and this is actually for the moment our maximal capability because of the color code recognition.
Mm-hmm. And these are natural cells, or can you use iPSC-derived cells, or what are your plans there?
Yeah, so at the moment we already use, of course, cancer cell lines because it's our first model, oncology. We also have primary cell lines, so we evaluate the hepatotoxicity on human primary. We have cardiomyocytes derived from iPS cells, and we also use immortalized cell lines.
Good afternoon, everybody. My name is Gianvito Grasso, and I'm the founder and CEO of Virtuo Labs. Today, there is more than 400 million people globally affected by rare diseases. It's more than cancer and Alzheimer's disease combined, and almost no one is getting a treatment. This is the stark reality we face today. And why is this happening? Well, it's because creating new medicine, as you all know, is incredibly hard. It takes more than 2 billion Swiss francs and 12 years to bring a single drug to market. And that's why rare diseases are neglected. The costs are too high and the process is too risky. But fortunately, artificial intelligence and generative AI are going to disrupt the way we currently design drugs. In our company, we have developed a computational discovery platform that combines generative AI and molecular simulations. Our platform is the result of tens of years of research and development in this field, where we combine award-winning machine learning models for drug design. We address the main reason why the impact of artificial intelligence in this field has been relatively limited so far, which is the scarcity and low quality of data. We can train our machine learning models not only on experimental tests, but also on synthetic data that we can generate using physics-based molecular simulations. Instead of physically testing thousands of compounds in a wet laboratory, we can virtually test billions of them. Our technology can predict which molecules are likely to work, and more importantly, which one will be safe. And thanks to our solution, we can increase the success rate of drug design by more than 100%. And this will make it feasible to address neglected diseases that affect hundreds of millions of people globally. Our business model is focused on the development of novel medicine, which allows us to retain IP to be out-licensed after the preclinical phase. We already have active drug discovery projects on neurological disorders, cancer, and metabolic disorders. And within the next few months, we are going to release our first computational discovery platform focused on nuclear receptors, responsible for more than 100 different pathologies. We have an amazing team of machine learning researchers and experts coming from pharma companies with more than 30 years of expertise in preclinical development of drugs and venture building. We already started active projects in collaboration with pharma companies, and we have just closed our first financing round of 2.7 million Swiss francs to advance our proprietary drug discovery projects focused on neglected diseases. So if you are interested in our venture, or if you want to speed up your drug discovery process, I'll be happy to tell you more about our company. Thank you.
Thank you very much. Any questions from the audience? Because otherwise, as an investor, I really need to know how you are different, for example, from Schrödinger, who is a big name in the space and claim the most advanced methodologies.
Yeah, they started more than 10 years ago, and they started mostly with physics-based molecular simulation. So currently they also have their own drug discovery projects, but they mainly have this computational platform based on physics-based molecular simulation. But then now we have seen the generative AI revolution, and most of the companies in this field, and we are talking about the preclinical licensing deals in rare diseases, most of these companies started before the generative AI revolution, and we are leveraging this revolution. So we are able to integrate generative models, chemical language models within our platform. And this is amazing because we can generate molecules tailored for specific pharmacological targets. So I think this is one of the biggest advantages that we have.
So this means that you have really seen kind of de novo molecules which
Absolutely.
are outside of the chemical space?
Yeah, absolutely. This is the biggest advantage of using chemical language model, being able to explore the huge chemical space. And that's the reason why generative AI is really going to disrupt this. And we can also integrate in this kind of chemical language models a lot of properties, for example, toxicity. And I think this is really huge.
Okay. And just a finishing question. Of course, the conventional chemical space is there because this was a result of a practical synthesis. So can you synthesize your molecules, which your computer is actually producing?
Absolutely. And this is the main limitation of the application of chemical language models. And that's why when I said the conditional generation, which means that we can train our model based on the synthetic feasibility and chemical feasibility, I think this is a big advantage, and this is what we did in our proprietary chemical language model.
Yeah, so hello everyone, and thank you for being here. My name's Kelvin Stott. I'm the founder and CEO of Amporin Pharmaceuticals based in Basel. Now, our vision is to effectively cure multiple degenerative diseases with a simple pill. And we aim to do that by a new approach by protecting and repairing cell membranes from the acute damage caused by protein misfolding and aggregation. So collectively, at least 50 different diseases are associated now with protein misfolding and aggregation. Collectively, these affect over 500 million people at a cost of $3 trillion a year and are growing rapidly with the aging population. And yet there are no effective disease-modifying treatments for any of these diseases. The unmet need is huge. Now, what all of these diseases have in common is that they a specific protein or peptide, sometimes more than one, aggregates, misfolds and aggregates ultimately into insoluble plaques fibers. And for years, the pharmaceutical industry has been developing antibodies, other therapies targeting these so-called amyloid plaques. And again, this approach has completely failed. Billions of dollars spent and gone. Actually, this is the smoke, whereas the fire, it turns out, is an intermediate in the aggregation process, because the oligomeric aggregates of each of these proteins are extremely toxic to cells at nanomolar levels. You put the oligomers on the cells, and the cells die. And the reason why is because all of these proteins form ion channels, pores where calcium can leak through. This is like puncturing a bicycle tire. And while pharmaceutical companies have been focused on the soluble oligomers now, this is like clearing the glass from the road. It's a huge effort, and it's only preventative and partially effective. Our unique approach to prevent the downstream effects of this is to specifically target the amyloid pores. And we are the only company doing this. Small molecule drugs to prevent the acute effects of these toxic proteins. Now, our results in vivo have been quite spectacular. We've shown with a single acute oral dose, a complete reversal of the neurological deficits in 2 established mouse models, Parkinson's on the left and Alzheimer's on the right, with no evidence of adverse effects in healthy mice. We aim to build a complete pipeline of drug candidates to effectively cure these diseases, ultimately leading to licensing after phase 1B or phase 2 trials. Essentially, we can do this rapidly if we're looking for an acute effect of disease reversal. We don't need big, long trials. And we aim to do this pretty quickly. So this is the team. Each of us have, from industry experience, each of us have got at least 20 years of experience in big pharma, biotech startups, as well as venture capital consulting. And we're very complementary. Daniel with his was head of translational medicine and neuroscience at Roche. Hervé in preclinical neuroscience, and myself, I'm a structural molecular biologist, more of a chemist by training. And we are looking to raise 7 to 10 million seed funding. So far, we've raised just 250K in convertible loans on the back of putting in 100,000 of our own money into this. Clean IP, we are 100% owners of the company, and so no issues with complexities of dead weights in the cap table or royalties due to tech transfer offices and so on. So that's our proposal, and thank you.
So at brainQr, we're developing groundbreaking treatments for major human diseases. And how do we do this? We tackle one of the biggest challenges in drug discovery. So what is that? It's intrinsically disordered proteins. Right? Disordered proteins have been previously deemed undruggable for many, many years, called like the holy grail of drug discovery. But disordered proteins play key roles in neurodegenerative diseases and in oncology. Our breakthrough AI-IDP platform makes disordered proteins druggable. So compared to conventional drug discovery, which has struggled for many years now to target disordered proteins effectively, our platform enables the rational design of small molecules which stabilize the disordered proteins in their native state, preserving functions. By leveraging AI and cutting-edge structural biology, we thus are able to unlock novel therapeutic opportunities for diseases with high unmet medical need. So we are advancing our strong pipeline focused on neurodegeneration. Our lead candidate BQ141 is a first-in-class tau stabilizer for Alzheimer's, and it's in preclinical development. In addition, we're developing a tau stabilizer for progressive supranuclear palsy and an AI-IDP stabilizer targeting the intrinsically disordered region of TDP-43 for ALS. Our goal is to advance these promising assets rapidly through the development pipeline in order to deliver transformative therapies to patients. So what makes BQ141 a game changer in Alzheimer's disease? BQ141 is the first-in-class small molecule which stabilizes tau intracellularly. It has demonstrated unprecedented efficacy in preventing seeded tau aggregation in neurons, which is the core pathology of AD. BQ141 is orally available. It has excellent blood-brain barrier penetration and superior safety. And I think what's for the business model, what's important, right? We can talk about biologics or antisense oligonucleotides, but being a small molecule, it's really a cost-effective solution with a very strong competitive advantage in this highly competitive space. Now we have a clear roadmap to bring BQ141 to the market. So currently we're in the preclinical stage preparing for IND-enabling studies. Phase 1 trials are set to begin in 2027, followed by phase 2 in 2028, and the pivotal phase 3 trial in 2030. With this market trajectory, we aim for market entry in 2033, providing patients the first disease-modifying Alzheimer pill. So we are currently fundraising and seeking €6 million to advance BQ141 to IND submission, and also to support our PSP and LS asset. We're also looking for strategic partnerships for co-development. Let's work together and make this happen. Thank you.
Thank you very much, Markus. Questions from the audience, please? So maybe just one question from me. You painted a very straightforward way to how to develop this in the clinic. Did you think about biomarkers to just make this process visible and to control dosing, for example? What are your thoughts about that?
Yeah, thank you very much. I think this is really a key question, right? And I think that's really what distinguishes us today on 27th of February from 5 years ago, right? I think now we have biomarkers which can be used to stratify patients, and in specific, for the case of tau, it's the tau biomarkers which really now provide the breakthrough in clinical trial design in monitoring efficacy, right? And this particular p-tau in the brainstem, where you select patients, you have to have a certain concentration, and then on top of that, you have p-tau 270 in the plasma, where you can longitudinal follow intervention, and also have a PD biomarker for readout. So it's a complete sort of change in the space there. Yeah.
So good afternoon, everyone. I'm Niccolò, co-founder and CEO of MicThera. And I know we think ourselves as a human, but we have at least the same number of bacterial cells in and on us. And these bacteria are not passive riders because they are implicated in a multitude of physiological and pathological conditions. However, clinical translation of what we see in the preclinical setting is really challenging. And in fact, several companies that are developing colonizing and grafting treatment fail to reach clinical milestone, leading to companies and program shutdown. And at MicThera, we take a radically different approach. Instead of developing colonizing and engrafting therapy, we develop and we mine gut microbial datasets to develop a new generation of molecule with an untapped therapeutic potential. And our platform combines advanced machine learning with screening technology to generate a list of potential candidates that are then synthetically produced and tested into preclinical mouse model. And now we show you our lead oncology program in prostate cancer, that because we have demonstrated that the prostate cancer is the gut microbiota is a key mechanism of resistance in prostate cancer, and we have a specific signature of bacteria that is associated with a better therapeutic response. And if we take this bacteria, we administer in top of standard care to preclinical mouse model as a live biotherapeutic product, we are able to delay the emergence of resistant phase. But at MicThera, we don't want to develop live biotherapeutic products, so we have mined the signature of responsive patients. Through our platform, we have identified two bioactive molecules, candidate P29 and 48, with a strong anti-cancer effect without off-target effect in normal cell. And we have also tested our lead compound in preclinical in vivo model of prostate cancer, both hormone-sensitive and cancer-resistant model, and by adding our molecule on top of standard of care, we are able to see a very nice tumor-controlling effect. And we are a multidisciplinary team with both key opinion leader in the scientific side, oncology space, and the business expertise. And so at MicThera, we develop microbial-inspired but synthetically produced molecules, and we have a platform that is able to unlock microbial-inspired potential, opening possible avenues of expansion in other therapeutic areas. And since our first pre-seed round, we have made progressive advance, and we have a lead candidate for cancer with prostate cancer as a primary indication, and we are doing a discovery phase thanks to our platform into E2 leads for infectious disease, because we are finding peptides that are active against antimicrobial drug-resistant pathogen. And to move our activity forward, we are seeking for 6 million investment by Q3 2025 to bring our oncology program in prostate cancer until END approval, and to get a preclinical proof of concept in vivo for our candidate in the antimicrobial space against multi-drug resistant bacteria. And so thank you very much for your attention, and I'm open to questions.
Thank you, Niccolo. Questions from the audience? Because otherwise I need to ask you, what do you think about the molecular mode of action of your bacterial product?
Yes.
How does this work?
So we have, let's be focused on the oncology program. We
Yes.
have a direct effect of our molecule on tumor cells, both resistant and sensitive to androgen deprivation. And in fact, we have done all our screening assay before moving to in vivo setting directly in vitro.
So direct tumor effect. Have you ever considered whether your bacterial products are affecting also the immune cells? Because, for example, gamma delta or MIT T cells are
actually
activated.
our lead program PV048 is not going through the immune system, because we have found that it's directly binding to some receptors that are present on the surface of prostate cancer cells.
Yeah, thanks everyone for sticking around for our little gem, Mighto Therapeutics. My name is Herman, I'm the CEO, and I'd like to present our disease-modifying treatments for mitochondrial disorders that we are building in partnership with Mitocon, a patient advocacy group in mitochondrial disorders. As you all know, mitochondria are the powerhouse of our cells. They generate ATP via this process. It's called oxidative phosphorylation, short OXPHOS. And that provides us with the energy for physical and our mental performance from birth on until our late life. And importantly, mitochondria rely on this little piece of DNA, mitochondrial DNA, that if it gets mutated or deleted, leads to neuro or muscle disorder due to loss of OXPHOS. A puzzling phenomenon actually in this context is that mutant mitochondria are, once established, actually manifest and proliferate so that they outcompete on the long run in the wild-type mitochondria and cause the disease due to a lack of energy. And we're harnessing new insights into the mechanisms that drive this expansion to reverse it by therapy. Here you can see two pieces of, two data points here from our compounds. One is here showing that our compound inhibits, our compound reduces the mutant load in patient cells and brings down the mutant mitochondria. And that actually leads to a recovery of respiration, which is a proxy for oxidative phosphorylation. We want to take these compounds first into a genetically defined group of mitochondrial myopathy patients, then expand into MELAS, the biggest mitochondrial rare disease, before going into Parkinson's disease, one of the neurodegenerative disorders that is clearly driven by OXPHOS defects. And we have all of these diseases have a really high unmet medical need for disease-modifying therapy and represent billion-dollar markets. We have a pipeline with a compound that's ready for a phase 1 trial, phase 1b, in patients, in mitochondrial myopathy patients. We have a second program that is discovery stage in Parkinson's. And to execute on this work plan, we're collecting a seed round of 10 million. On this journey, I'm working together with my business partner, Niccolò Bacchi, and we have an excellent team of researchers from Europe and the US that provide assets in the IP that we have in the company. And we are accompanied by our patient advocate, Piero. Taken together, Mighto is developing disease-modifying drugs with disrupting potential, attacking mitochondrial disorders upstream of all other approaches, mitochondrial rare diseases and Parkinson's. We have a small molecule pipeline and clinic-ready assets for phase 1b to collect a seed round of 10 million. Thanks for your attention. I'm open for questions.
Thank you very much. Any questions to Hermann? Mitochondria and mitochondrial myopathies are also having an inflammatory component, since very often the mitochondrial DNA is released and activating inflammasomes. So how do you see the role of inflammation intervening with your approach of treating these myopathies?
So it's a really interesting perspective, because this innate immunity component is just popping up as a major theme of mitochondrial biology, and we are trying to restore mitochondrial functionality by really intervening at the earliest point. That's when the disease emerges, and we're preventing the mutant mitochondria or dysfunctional mitochondria from expanding further. So we're actually quelling it at the first stage.
So this means that you are going after pediatric patient population?
We will, on the long run, but we have to establish safety first in adults and then go for a rare pediatric disease designation, yes.
Hi, everyone. Thank you for the introduction. My name is Elsa Brio. I'm a CEO and co-founder of PannTheraPi, a French biotech company at clinical stage developing innovative products in neurology by acting on a new specific target. The company was founded based on innovation coming from Collège de France in Paris by two scientists. One of them is also a neurologist specialized in epilepsy. I have a PhD in neuroscience and more than 15 years of experience doing the cross-development of new products in neurology and rare diseases. And we have a great team, a small team, but we are still agile. The company have 4 years now, and we obtained more than 5 million euros, but only a pre-seed with private individuals and more than 4 million as non-dilutive, and especially we obtained the EIC accelerator. We also have equity available up to 10 million with the EIC. Our first product is developed in epileptic syndrome, which is an orphan disease. And as mentioned, we act on a new specific target, which have been found using postoperative brain tissue from the patient. Our scientists demonstrate that ex vivo, the tissue continue to have some seizure, so we are able to directly screen the molecule on the tissue. The advantage of this target is that it's fully specific. It's only overexpressed and also activated under pathological condition. And by blocking it, we are able to fully stop the seizure in 80% of the cases, with a stronger safety because we have no impact on other cognitive function, on the contrary of other anti-epileptic drugs, which have a lot of safety issue. We are doing our development with the first product, which is a repurposed, reformulated drug, allowing us to have a very fast-track development and also a safer product. This is a product, and we are currently at clinical stage with this product, but also as a target is of interest for other diseases with crises. We are developing new chemical entities which could be developed in other diseases of interest, such as multiple sclerosis, pain, but we are also talking about neurodegenerative and psychiatry. Using our unique platform, we have post-operative tissue from patient among others, but also AI and specific in vitro model. Currently, our first drug is at clinical stage. We already done the phase 1 clinical trial, demonstrating, of course, the safety of the product, but also the long release profile of our pediatric formulation. We also identify some biomarkers which are linked to the target engagement. We are ready to start a phase 2 trial in a small group of patients, and we already discussed the development plan with EMA, and it's ongoing with FDA. On parallel, we would like to pursue the development of our new lead. And the clinical data for our first product will validate the target and our platform. For that, we are looking for 10 million seed funding. And as mentioned, we already have more than 50% of the amount with the EIC and other VCs. It's a de-risking first product. We have a unique platform and already 5 pharmaceutical companies under CDA looking for clinical data with our first product. Happy to pursue the discussion, and thank you for your attention.
Thank you very much, Elsa. Any questions about that? So, yes.
Hello, Martin Heuber. Sequence, is this a small molecule drug? Or?
Oh, yes, sorry. It's a small molecule and it's oral drug, so you need to take it 2 times a day.
Okay, thank you.
And how do you see, you mentioned this can be still an orphan disease, but epilepsy is broadly treated, and just a couple of days ago there was also a transaction on Dravet syndrome, again focusing on that kind of disease. So how do you see your position within this very broad field, especially against very well established OTC plain drugs?
Because we are not targeting Dravet and not Lennox, it's another syndrome. Epilepsy is not a single disease. There is a lot of type of diseases. And our first indication, currently there is no competitor, no drug under development. So we do not need to have a comparator during the clinical trial, for example, and it's a market of 2 billion. And we obtain post-operative from those patients. So we identify that our target is overexpressed in all those patients, whatever the age, the sex, and also the onset of the disease. And also we have some literature data demonstrating the link between the target and the number of seizures.
So hello everyone, I'm Tiina Kähkönen, Chief Scientific Officer at OncoBone Therapeutics, and we are developing therapies for cancer bone metastases. So bone metastases are a major problem in many cancers, and they affect 70 to 90% of advanced stage breast and prostate cancer patients. Only 5% of these patients are alive 5 years after diagnosis, and bone metastases cause a massive bone destruction and bone pain, and therefore significantly decreasing the quality of life. Also, there are no curative treatments available, making bone metastases a high unmet medical need. Within the team, we have special expertise in bone metastasis research, preclinical modeling, and drug development, and we established OncoBone Therapeutics to develop the first safe and effective therapies for bone metastases. In our business, we are using 2 innovative concepts, which I will describe you in the next slides. Osteoimmuno-oncology is our concept to identify therapeutic targets which are expected to have the best possible efficacy, but also bone safety. Due to this target identification and the development in our AI-based platform, we came up with our lead asset OPP-004, which is a novel small molecule dual inhibitor of CDK9 and CDK13. It has some special benefits, so it is highly selective and has no activity outside the CDK family. It has a high tissue penetration to brain, lungs, spleen, and kidneys, and 17 to 22 hour half-life, allowing a long-lasting oral exposure after a single oral dose. It has a specific mode of action which suggests some benefits over other competing CDK9 inhibitors. Bone metastasis technology platform is our way to reliably study the effects of therapies in bone, specifically on bone metastases during the preclinical stage of research. When we subjected OPP-004 into this model system using a triple-negative breast cancer model, we did see a significantly reduced tumor burden, bone loss, and also bone pain, suggesting that this could be a potential treatment for patients with bone metastases. We are currently seeking to raise 1.5 million seed funding to continue development of OPP-004 into a dual treatment with anti-tumor and bone protective effects. And we are currently considering on enhancing the effects either by doing combination treatments or developing OPP004 further into an antibody-drug conjugate. This seed funding would be used for confirming efficacy, and it would be followed by a Series A for enabling completing the IND-enabling studies. And after that, we would be looking for an out-licensing partner for clinical development. So thank you for your time and attention, and happy to speak with anyone who's interested in our program. Thank you.
Thank you very much. Questions from the audience? Can you give us a little bit more detail on your lead program? So I, of course, have the advantage of reading your leaflet, and it says this could be a CDK9 inhibitor. Is that correct?
Yes, so it is a dual inhibitor of CDK9 and CDK13. So it is a novel target. There are some other CDK9 targeting inhibitors currently in clinical development, but ours is the first targeting this dual pathway or dual CDK9, CDK13 inhibitor. And because of this dual aspect or dual targeting, we can actually see an enhanced or improved efficacy, especially on metastases and bone metastases. So that is the reasoning behind of this choice of indication that we are going for.
So previous experience with CDK9 inhibitors revealed some unwanted effects as well.
Correct.
So what do you estimate? Where would you end up with that?
Yes, so the previous CDK9 inhibitors in the development, some of them have failed, and that has been due to that they affect really a broad spectrum of different CDKs. So that's why we are going towards the more CDK9 specific inhibitors. And now with this second generation and with our asset as well, we do not see that kind of a classical adverse events or anything that was seen in the earlier development, suggesting that this could be even a safer approach. And of course, it provides this high efficacy on tumors. So that's why it's a good treatment option also for more advanced stage patients.
Thank you. Time starts, okay. The future of biologics is not simply finding new drugs. I think as Kelvin and Marcus mentioned, it is all about the target. The real culprit, the target, is the most important point. That's where we are reimagining the drug. We are reimagining the antibody using AI to target only precisely those culprits, the proteins that misfold and cause the disease. So we have a platform company where it has more than one disease indication. We are working on protein misfolding precisely, exclusively. And the challenge, as I mentioned, thanks to them, they introduced a lot about protein misfolding. So this is now appearing to be a real target for the pharma, which was a blind spot for a long time. It was like a fight club. Everybody speaks about it, but nobody knows and nobody sees that problem. Because we all were interested in targeting oligomers or proteins that misfold, but we can't see them. It's not visible, and you need to visualize it in order to make that impact. So that's where we bring in the natural antibodies. Thanks for making it easier for me, Thomas. Natural antibodies that are existing in our body that prevent us from developing these kind of diseases. And we use AI, the deep learning method, to tweak them in a way that it becomes better antibody in terms of the binding and the affinity and also the solubility and other properties. So we have the right animal model, which is the canine model, and we have a lead candidate, and we have an AI algorithm that can kind of visualize the oligomer structure. And that's where we are pushing the project forward, and we are raising 6 to 8 million. And to advance the Alzheimer's and non-CNS disease indication, the second one, and for the animal models. So I think thanks to their introduction, it's very obvious it's not only in the brain, the protein misfolding happens everywhere. And we also can translate into different disease indication, including oncology, or name a new virus, and you also can have the structure and then predict from there. And yeah, just to super simplify Alzheimer's, whenever you target monomeric form, the amyloid beta, it is inefficient. Whenever you target the aggregated, the stacked version, it's like the single leaflet of paper. That is the monomer. Stacked version is the pile of paper. If you touch this, it will not be effective. The touch that is an adverse effect. And that shows to approval FDA, you got it for the same amyloid beta, and then only problem remains, oh God, time flies, sorry, is the adverse effect, which is the ARIA. And our antibodies do not bind to amyloid beta as monomers or fibrils. only to the oligomers. So we have a cold fusion AI and natural antibodies, and we deliver them to prevent the disease from developing further. This is the great team, fantastic scientific advisory board. Market, you know, I don't need to tell you. And then these are the next steps, preclinical studies. Thank you so much for your attention. Start stopping. It's minus something.
Okay, so let's start and try to keep that in 3 minutes time. Thank you, Thomas, for the opportunity. Thank you, everybody, to stay in the room. My name is Frederic Fasano. We founded a biotech company 1 year ago called Alpioner Therapeutics, basically on a bacterial vector technology that we licensed from Grenoble University. We have a team of experts, basically 2 scientists working respectively in vaccine development, Klaus Schwamborn, based in Düsseldorf, and Rémi Palmentier, a specialist in immuno-oncology. My background is I'm a pharmacist by training, and I led several programs, but late-stage clinical and commercialization of product. Our vision is to unleash the potential of this innovative bacterial platform with 2 ideas behind, one in infectious disease, developing a vertical in infectious disease, and another one in immuno-oncology. Basically, what we are doing is using a bacterial construct, which is a secretory system, so-called the T3SS, T3 secretory system, as an injectisome, basically as a needle to inject protein of interest directly into the cytosol. The benefit of doing so, of course, we are working on one side on the bacterial vector, which is genetically modified and then transformed in an aberrant bacterial vector, and we are loading also, this is the software part of the technology, we are loading a plasmid in order to code for some peptide of interest. This needle is particularly interesting in its ability to inject high quantity of peptide of interest. Basically, as I said, we have 2 verticals today. Basically, on changing the load of the plasmid, we can program the platform against certain pathogen, multi-resistant pathogen. One of them, this is our lead candidate. It is a vaccine against Pseudomonas aeruginosa. And the second lead program we have is a personalized therapeutic cancer vaccine based on neoantigens. We have a certain level of demonstration of the platform in its ability to be efficient in the tumor growth. This is a well-established tumor model based on ovalbumin. So basically, the construct is injecting, so is working on the ovalbumin antigen, and then we measure in this well-established model the impact on tumor growth. What we see that the tumor growth is substantially reduced, and it is comparable. I need to speed up. It is comparable to what we have seen with mRNA technology. We have the same proof of concept for the anti-Pseudomonas vaccine. What we are seeking here is basically 5 million euros that we can match with non-dilutive fundings in order to bring the 2 assets into a clinical stage. So we have to work on both sides, and we'll need probably 18 to 24 months to bring those 2 assets to a clinical stage. Mr. Chairman.
Thank you very much, Frederic. Questions from the audience? Now, I have to ask you a very different type of question.
Please.
As a Basel-based investor, we are very much aware that T3 Pharma was acquired by Boehringer Ingelheim 2 years ago. This is obviously your competitor, and watching the competition is very important for startups. So can you just share your opinion on that?
T3 Pharma, I mean, is our own benchmark, of course. We know inside out, I would say, this company. For us, it's a validation of the approach. Many new technologies have been emerging with huge promises, but again, for different reasons, we don't have time to explain today. The bacterial approach is providing some very interesting insight into level of efficacy through the immune system and so on and so forth. And what T3 did actually is validating the approach. Now, the difference is they are not using exactly the same vector. And we are in a mode whereby the plasmid that we use, the plasmid is inducible. So in a way, it can be stimulated to increase the release of those peptide of interest and the quantity of a peptide of interest into the cytosol. So we think we have some interesting, specific feature to our platform. But of course, I mean, we look at T3 as our benchmark, and hopefully in 5 years' time I could stay on stage and say, yes, we have been bought for the same amount of money.
Thank you. Hi, so I'm Nick. I am the co-founder and CEO of Serenatis Bio. We are a new biotech based in Cambridge in the UK. My background is drug development. I spent the past 20 years developing a drug through preclinical, clinical, all the way to regulatory approval and into the clinic. And I'm now using that expertise to focus on obsessive compulsive disorder. So we have launched a pre-seed round of 1.3 million. We're already at 1 million committed investments. We're looking for investors to help us reach the target. So OCD, it's not about lining up your pencils. It's not about color coding your spreadsheets. It's a devastating mental disorder. I've had it for about 35 years, sometimes very seriously. I run a support group in Cambridge of people with OCD. And my best friend 5 years ago killed himself. He had a form of OCD called false memory OCD, where his brain would make up false memories of horrific events, and then he would spend his days obsessing about had he done it or had he not done it. It was absolutely horrible. So it's a very common disorder, around 3% of the population at least. That's 250 million people worldwide. And current treatments, there's been no new treatment since the early 90s. It's very high doses of SSRIs, 5 to 10 times the antidepressant dose. So a huge unmet need, but also a huge commercial opportunity. And we've calculated our first drug could reach peak sales of 4.5 billion. Okay, so we're working on 3 different drugs. The first one is an mGluR2/3 agonist, and we have worked on this with the University of Cambridge over several years. Okay, and so we are now going into preclinical work with this. If the preclinical work is successful, we can go straight into the clinic. And this redresses the glutamate GABA ratio, which is at the core problem driving OCD symptoms in the OCD brain. Our second drug is a drug that my co-founder, David Kavala, who is a chemist and a neuroscientist, has been working on. And this is a dual action AMPA glutamate and mu-opioid modulator that is based on known mechanisms of action within OCD. And we have recently shown that it works really well in preclinical models. And our third drug comes out of work I've done with the University of Chicago with Professor John Grant on COMT inhibition, which affects dopamine in the prefrontal cortex and which we have shown reduces OCD symptoms. Unfortunately, the only COMT inhibitor that's in the clinic has liver toxicity, so we are developing new ones that do not. We're using precision psychiatry, particularly EEG biomarkers, which the University of Cambridge has shown are linked to glutamate GABA ratios and can help us select who will respond to the first 2 drugs. And for the third drug, there's a saliva test where you can test for a mutation to see who will respond to it. So this is the team, myself and David as the co-founders and our advisors from Cambridge and also from Chicago. So we're raising 1.3 million. We hope to reach that very soon because we've already raised 1 million. We've already started the work because our lead VC has already advanced funds, and we're going to do a much larger round towards the end of the year of around 20 million to go into clinical trials. Thank you.
Thank you very much, Nick. Questions from the audience? Can you detail a little bit your developmental plan? Because you have now 3 or 4 drugs, each going for a certain target. Do you think there will be a response to each and individual molecule in a certain patient group, or do you plan to do combination therapies at a certain point? How will you actually construct a clinical trial for that?
That's a very good point. So we've already got plans for our phase 2 study, which will be for the first drug, which will happen next year, which will be a crossover design. So they're in order of importance. The first one can go first into the clinic. The second one goes into GLP tox. The third one's at discovery stage. Now, in psychiatry, no drug works on all patients. And that's why we bring in the precision medicine approach to help identify who should respond to the glutamatergic drugs and the COMT inhibitor. But on combination therapies, it's an interesting one because the COMT inhibitor works on dopamine dysregulation. That helps with cognitive flexibility, okay? Whilst the glutamatergic ones are more with the compulsivity. So yes, there are opportunities for drug combinations later on, absolutely.
Okay, hello, my name is Kaido from Vectiopep, and we enable antigen-specific T-cell activation with our technology for clean messenger RNA delivery very specifically into the immune cells in patients, and we look into applications for this in oncology and autoimmunity. the classical cancer-targeted therapeutic approaches have the advantage of targeting many different patients. The downside is that the clinical response offers only a tiny increase in survival rates. On the other hand, immune checkpoint inhibitor type of approaches in immuno-oncology offer excellent clinical results, and the only challenge there is that the target patient group is only a tiny subgroup of the patients. So any approaches that would allow us to widen the patient selection who would be responding to the therapy with excellent clinical results would be really welcome and important. And one way of achieving this would be looking into new therapeutics, new approaches, and achieving antigen-specific T-cell activation, which would allow us to target even the patients with so-called cold tumors. And this is exactly what we are aiming in Vectiopep. So how do we achieve this specific T-cell activation? Well, it is well known that the mRNA or messenger RNA delivery with lipids or LNPs are very efficient way of transporting the material into the liver, which is a very nice way of targeting liver diseases. In Vectiopep, we have developed an alternative messenger RNA delivery, which allows very selective transport into the lymphoid organs, such as the spleen, while practically completely avoiding the off-target accumulation in the liver. And when we look closer into the cell types that we are actually delivering into or transfecting, it is the antigen-presenting cell types, the dendritic cells, and the macrophages. So that offers a very clever way of activating the T cells and inducing specific manipulation of the T cells in the patients. And one key aspect in activating the T cells is also very a specific activation of certain immunocytokines. And we are inducing transient but very strong activation of certain immunocytokines, which the LNPs are not doing. This is the proof of principle or proof of concept animal model tumor treatment. Actually, two different models. One is colon carcinoma and the other is melanoma. And in both of those aggressive models, we are showing that with our peptide technology, we are able to strongly control the tumor growth. And we are also outperforming the lipid-based approaches. We are taking this into two clinical programs, one advanced melanoma and the other colon carcinoma, but we are also looking into autoimmune diseases, because this would be a nice way of utilizing the specific manipulation of the T cells. The chemical composition of the Vectiopep technology is peptide-based, as the name says, so it's cell-penetrating peptide-based. And our team is very small, but looking for ways of expanding. And if you find this technology interesting, feel free to reach out and communicate. Thank you very much.
Thank you, Kaido. Questions from the audience or from me? So I just noticed that you are going after microsatellite stable colorectal cancer, which I thought is interesting, because the conventional wisdom in anti-tumor immunity is that the mutational load is proportional to the response rate. So the more mutations you have, the higher chances you have. And microsatellite stability is probably restricting that. So what was the rationale behind this?
That is a challenging question indeed. There are arguments to be made, but our main strategy is to have a specific T cell activation, which would also allow targeting so-called cold tumors.
Okay. Good afternoon, everyone. It's a pleasure to be here and share with you our vision for Procavea Biotech, which is a recently incorporated ETH spin-off. And we leverage a unique protein-based technology to deliver on the promise of RNA medicines. My name's Tom Edwardson. I'm the founder and CSO. To give some context, I'd like to start by looking at RNA medicines that are on the market, specifically siRNAs, for which there are currently 6 approved therapies. Now, these therapies are for rare and common diseases. And the reason that there are not more therapies on the market based on siRNA is not any inherent limitation of the technology itself. siRNA's general mechanism of action is what's so exciting about it. But we lack the delivery systems. So of the current approved drugs, the truth is that of the injected dose, less than 2% reaches its biological target, and we can currently only reach targets in the liver. So clearly there's room for improvement and there's a need for innovation. And that is exactly what Procavea is offering. So we've developed a unique proprietary delivery system based on computationally designed non-viral protein cages. We produce these monodisperse hollow particles from a scalable production process, and then they can be mixed with any RNA for a different disease target to create a stable, well-defined drug carrier ratio product, which we can then, in this case, in this example, deliver to the kidney. So with a systemic injection, we can reach the kidney. And what's really important here is that by modification of these particles with an endosomal escape domain, we can overcome the limitation of endosomal entrapment, which is really the root cause of this low efficiency that I mentioned earlier for most RNA delivery systems. Based on our initial in vivo data, we aim to focus on developing 1 to 3 drug candidates in the area of adrenal or kidney disease over the next couple of years. And we're looking now for a seed round of 3 million Swiss francs to help us finish the preclinical development. This will then be followed on by a larger Series A, which will depend exactly on our lead indication and the clinical development plan. We have a platform, and of course, in parallel to this, we're looking for opportunities already in partnering in different disease areas. At this early stage, we're lucky to have a dedicated team with deep expertise in protein engineering and siRNA therapeutic development, as well as experience in biotech startups. We also have really good support from the Indo-Swiss coaching system. And with that, I'd like to invite any of you out there, especially early stage investors, to reach out to us if you can see yourself being part of our vision to deliver on the promise of RNA medicines. Thank you.
Thank you, Tom. So since you were asking about investors, so from an early investor perspective, a platform is always interesting, as we heard before, but we like assets. So can you give us a little bit more detail on your kidney thing? Mainly,
yes.
How did you achieve kidney targeting? Why not everything else? And what is actually your real target within the encoded part?
Yeah, this is to be honest, in terms of a disease indication relevant target, we're exploring this right now. So I say area of kidney or adrenal. How we get our particles there is due to the size of the particle and the surface chemistry, let's say. These are also compatible with cell surface receptor targeting, and we've validated that. But we're now evaluating, with the help of kidney experts and adrenal experts, what are the best diseases with a validated genetic target that's amenable to siRNA knockdown. So that's what we're doing right now.
Okay, so may I assume that your particle size is about, let's say, 200 nanometers or something like that?
Much smaller. It's 14 nanometers.
14?
Yes.
It's a completely different range.
Yeah.
So why do not you then end up really in liver or phagocytic cells throughout the body?
That's a good question, and it's a long discussion. Let's say for now, I just say this is what they do. Okay, thank you.
Let's leave it for later.
Thank you, and thank you everybody for sticking around. As mentioned, Thomas, I'm Alistair Irvine. I'm the CEO of Scarlet Therapeutics. We're a spin-out from the University of Bristol with a co-founding team that has a strong mixture of scientific excellence and executive biotech experience with exits. So what we're about is pioneering the use of universal red blood cells. So I mean red blood cells that can go into anybody no matter what their blood type, in the unmodified form for general transfusion, or in the modified form for the treatment of disease. So why are red blood cells interesting? Well, they're inherently safe. A lot of cell therapies, you're concerned about tumorigenicity. Red blood cells don't have a nucleus and don't divide, so they can't form tumors. They're enduring. They've got a lifespan of about 4 months, so ideal for long-term treatment and very rare dosing. They're pervasive. They go everywhere in the body, so you can treat everywhere in the body, so you can target specific tissues or you can treat systemically, and you can make them smart. So you can engineer them to carry extra proteins so that they can perform extra functions as they're circulating, and those proteins can be either intracellular or within the membrane. So this is very much one platform with many applications, and we're initially focused on metabolic diseases on this slide. So one question you've got to ask is, if red blood cells are such good therapeutic vehicles, why aren't there products on the market already? And the challenges faced by the field so far have been really because the red blood cells have been generated so far from donated blood. And if you're using donated blood as a start of your manufacturing process, you've got a lack of scalable manufacture, a lack of reproducible manufacture. You need blood group matching between your donor's for your manufacturing and your patients. And there's also been challenges about loading the red blood cells with enough of the proteins that you want within them so that they can perform the function. We've put together a suite of technologies to address these issues. So first of all, we generate our red blood cells from red blood cell producing cell lines. And we have exclusive IP around this technology. So we have immortalized cell lines that will produce red blood cells. So we solved the scalable and reproducible manufacture issue. We are also able to generate universal red blood cells by very careful selection of the blood groups that are on the red blood cells. And we have technologies for maximizing the activity. So our initial focus from an indication perspective, as I've already said, is metabolic diseases. And to narrow it down a bit, metabolic diseases where the clinical effect is caused by a buildup of a systemic level of a toxic metabolite. And the concept is put into the patient red blood cells that will process that toxic metabolite into something that can then be excreted or used by the body. So it's really a detoxifying machinery circulating. We've got proof of concept in 3 indications. This is just data for hyperammonemia, showing that we can get rid of ammonia and generate an amino acid. And at the moment, we're doing a seed financing. We're looking for 5 million, essentially to move this technology into in vivo proof of concept to generate initial data and select our first product candidate, which to carry forward to the clinic. If anybody's interested in learning more, I'm very happy to talk to you about it. Thank you.
Thank you very much. Questions? So 2 questions, of course. You have a universal red blood cell. I assume the blood groups are kind of wiped out. But what about the rare blood groups, which still can use alloreactivity?
Well, essentially, a lot of the blood, what we do is most people think about universality. They think O negative. You know, obviously there's a lot more blood groups than that, and there's a lot more allo reactions than that. So what we do is we start off with a cell line that's selected so that it's as universal as possible over 8 or 9 different blood groups. And then any other blood groups that are problematic within that, we can knock out. Because it's a cell line generated thing, we can, as long as they're not essential, which most of them aren't, you can just knock them out by CRISPR technology. And we've got publications showing that we can completely wipe out a vast number of blood groups.
And I just assume during this procedure, generating your cells, these also undergo enucleation, so they lose their nuclei. So how do you then still ensure that they will be able to produce your therapeutic protein for a long time?
They don't produce the protein. Essentially, the protein that's in the red blood cell when it's first created in the manufacturing process, that's it. Just like hemoglobin is the protein in a red blood cell when it's created in the body, and then that cell is stable for 4 months.